Welcome, everybody. My name is Ben Garcia, President-elect of US HUPO, and it's a pleasure to be here as a moderator for this Spotlight on Trainees webinar put on by US HUPO. This is sponsored by the US HUPO Early Career Researcher Committee, and today you're going to be hearing from our next generation T-mass spec scientists who will share the research they presented at the US HUPO conference earlier this year. I think just a couple things. What we're going to do is have three talks, and after every talk, we'll pause to have some questions. And if you out there have a question, please just put it in the chat in the Q&A, or not the chat, the Q&A, and we'll be sure to kind of monitor that. And so without further ado, I'd like to introduce Leah Barney, who's from Vanderbilt University. Leah, it's all yours.
Thank you. Let me go ahead and share. 2 seconds. Right. Let's see. The share is not coming up. 2 seconds. Sorry. All right. So, just leave this far. Thanks. So the title of my talk this afternoon is Monitoring the Dynamics of the Endoplasmic Reticulum Stress Response Pathways in a Dual Species Co-Culture Model of Human Aging Neurons through DIA Mass Spectrometry. So to start, approximately 1/3 of the human proteome is trafficked to secretory environments. The endoplasmic reticulum is the major site of secretory protein processing and folding, as well as post-translational modification. To ensure the secretion of properly folded proteins from the endoplasmic reticulum, there is numerous chaperones, trafficking and folding factors and degradation factors, which act collectively and are known as the proteostasis network. However, when there's an increase in aggregated or misfolded proteins in the ER, that unfolded protein response, or UPR, is activated. UPR consists of 3 major branches: ATF6, XBP1, and PERK. And activation of the respective transmembrane receptors in the ER lumen results in the production of distinct transcription factors in the ATF6, XBP1, and PERK. And these transcription factors, after binding DNA in the nucleus, this results in a transcriptional and translational reprogramming of the proteostasis network to restore the secretion of properly folded proteins from the ER. UPR, whether it's overactive or underactive, is known to be involved in numerous pathologies, including diabetes, cancer, and age-related neurodegenerative disease. However, our understanding of how the UPR plays a role in these diseases is mainly hindered by the challenges that studying the UPR entail. The UPR is cell and tissue type specific. It also the degree of the response and how the response and the different effectors of the response can be influenced by the type of ER insult. It's impacted by temporal regulation, feedback between the different branches of the UPR, and also crosstalk with other signaling pathways, for example, the integrated stress response and also the oxidative stress response. So typically, transcriptomics is utilized to study UPR activation to measure the pathway. However, this does not get at the translational regulation of the pathway. Additionally, Western blot analysis has been utilized for a long time to mainly look at the core proteins involved in the UPR, so that looking mainly at the transcription factors and also the transmembrane receptors and if they're active. And the only issue with this then it becomes is how do we know what the other effector proteins are doing when the UPR is active? That's why we turn to proteomics, and this is what our group has largely used to look at the UPR. And today I will be talking about how we decided on new branch-specific UPR targets. So in order to identify branch-specific UPR proteomics targets, We've utilized engineered stable cell lines, which with activation using small molecules, we can upregulate the respective transcription factors that associated with each branch of the UPR. So after treatment with these small molecules for 16 hours, these cells are then collected and lysed, and they undergo sample processing for mass spec using our automated sample handler. These methods were developed along with my colleague, Jake Hermanson. They utilize on the biomech these SP3 beads for protein digestion and cleanup. These samples that were specifically generated using the engineered stable cell lines underwent data-independent acquisition analysis and also tandem mass tag data-dependent acquisition analysis. So looking at the difference between these two methods in the detection of UPR activation, we see that DIA is able to detect more peptides per minute. This results in a greater number of protein groups being identified. And there's a high degree of overlap between the methods, however, which makes this very effective for studying the UPR. So if we take a look at some of the previous targets that were devised using RNA-seq, and we look at how many of those targets, those RNA-seq targets, we can detect with mass spec, we see that DIA really allows us to get at more of these very low abundant targets. And that's really the challenge that the UPR, studying the UPR has also faced from an analytical perspective. So looking at the transition from these RNA-seq targets to proteomics targets, the genes highlighted that are bolded are those that were retained from RNA-seq, and then the unbolded gene names are the ones that we determined via our DIA proteomics approaches. And as you can tell, we've refined and expanded these lists, and this really highlights the difference between transcriptomics and proteome, and the difference between UPR regulation at the translational and transcriptional levels. So then if we take these targets and we apply these targets to then when the UPR is globally activated, we can do this pharmacologically using capsigargan and tanikamycin. We can see that we are able to indeed detect activation of the 3 branches of the pathway. So this is just a quick validation. In getting to the main, what I'm really trying to talk about here is now how we applied these targets, these proteomics targets, to study the UPR to this human co-culture dual species model. So this was done in collaboration with the Cavallari lab at Vanderbilt in the Brain Institute. And what they had found is that with age, there is this increase in action potential evoked desynchronized release. And they noticed in this in vitro model that with age, there's an increase in NMDA receptors. NMDA receptors are mainly present on the postsynaptic cleft of neurons and are highly calcium permeable. This was associated with them seeing ER stress, markers of ER stress from a Western blot perspective, and also downregulation of these key presynaptic proteins, SYT1 and CSP alpha. So with this in mind, we went ahead and thought, what if we look at the UPR from a global perspective instead of just doing a Western blot approach? We're only able to look at a few markers of UPR activation. So go into the model a little bit more. It's human embryonic stem cells, which are induced to cortical human neurons. These neurons are then grown in culture on a bed of glia. So those are 3 different cell types in the glia. They are then matured. The 30-day time point is considered when these cells have matured, and then they're aged out to 50 and 60 days. These samples are then collected for processing and undergoes our standard quantitative proteomics workflow for DIA. We think more about this co-culture dual species. We have peptides in here that will be human specific, mouse specific, and then shared. So in order to get a better sense of what's happening to the neuron specifically and the glia specifically, looking at quantification of these peptides that are specific to human and mouse is really important. So we went ahead and generated a theoretical triptych database. We see that about 30% of these peptides are shared, and another 30 are human specific, and another 30 are mouse specific. Utilizing Diane outputs from our DIA analysis and our species-specific triptych database, we can then match our observed peptides with our theoretical peptides to determine which of our observed peptides are indeed human species-specific and mouse species-specific, so that we can obtain this species-specific protein quantification. So in validating our data analysis pipeline, we made a set ratio mixed species model. So these are human and mouse lysate were mixed. These are neuroblastoma cells in differing amounts of human lysate and mouse. We can see, just as a quick validation, that when we increase the amount of human lysate, we get a respective increase in the percent abundance of these human proteins, as we would expect. And this ratio mixture has also allowed us to validate our code and also control the false positive rate. Thinking more about detection of the UPR in a dual species model, we developed another mixture of lysates, this time though with SH5Y cells that were activated with global UPR activators, as mentioned before, thapsigargin and tunicamycin, and mixing 80% mouse lysate with 20% human, with the 20% human either being treated with DMSO or the respective global UPR activators. We can see that we can detect UPR activation, whether it's a mixed species model, meaning both human and mouse lysate being mixed, or only human lysate. So this is just an important validation that we can actually detect UPR activations in complex mixtures. So now going back to our work with the Cavallali group, looking at now that we have a pipeline for human neuronal quantification of these proteins species specifically, we can see that when we look at the ATF6, PERK, and XBP1 pathways using the target proteins that I mentioned earlier, we see that at the day 50 time point, there's significant decrease in each of the pathways of these targets, whereas in the co-culture glial cells, which are mouse-derived, the opposite looks like it occurs at the day 50 time point. Some of them are significant, whereas ATF6 is not, but it's the reverse trend, which is interesting in itself. And then for our controls, we have glia-only controls, and those trend towards decreasing in these proteostasis factors that are regulated by the UPR. So in general, these changes basically highlight that the UPR is changing with age in these different cell types, but not in the same direction in both, which is interesting and something that we're definitely following up on now to kind of see what that's coming from. So in conclusion, we discussed that DIA provides more comprehensive. This light out in this room. Oops, 1 second. Yes, more comprehensive UPR target coverage. The dual species co-culture model can be used to understand how the UPR changes with age. Branch-specific UPR target proteins can also be utilized to study different cell types and disease states. So I want to thank my lab, Dr. Lars Plate, for constantly letting me try to do different things, continue to grow as a mass spectrometrist. I forgot in the slides, but Sarah Garcia played a very large role in getting these pipelines up and running for the group. And as I mentioned earlier, Jake helped me with a lot of the pipelines for the biomech. So, yep, and the Cavallari lab, those two students, Barack and Aiden, were very important in getting these co-cultures done and handed over to us for analysis. Thank you. I will take questions. I can hold on 1 second. Try and turn the
Thank
light
you,
back.
Aaliyah.
Sorry about this. Yeah, this is really,
Yeah,
yeah.
like you're telling ghost stories.
Yeah, it
Um,
seems that way.
is there any questions for Leah? If you do, please put them in the Q&A box. I can start while people may think about asking you a
Sure.
question. Kind of have technical and biological question.
Sure.
Um, in terms, yeah, it looks like your DIA approach identifies more targets.
Mhm.
Have you compared the quantification though of both? Because I always heard TMT should probably give you more, I guess, robust quantification than like label-free kind of approaches.
Um, so I have somewhat, I've done some correlation plots, uh, basically looking at the degree of the fold changes. And we definitely see that the fold changes are higher in DIA. I think basically because we do our TMT and MS2, we don't have a tribrid.
Oh, okay.
But
Yeah.
as far as missing values go, there I've definitely taken a look at that. Yes, for these specific samples, the missing values in the TMT are really low. There's a little bit more in the DIA samples, but um, when all is said and done, it doesn't look like that the loss of IDs that we get from the missing values seems to make that big a difference. I don't know if I'm answering your question
Yeah,
right,
you're
but
getting at it. Yeah, no, yeah, I think so. It's just kind of general questions on robustness. I think we do have a question in the question answer box. So I'll just read it off. Has co-culturing changed the depth measurable when you measure multiple species simultaneously? If there is high similarity between species, would you lose quantitative information on shared peptides?
Yeah, so I do see that, yes. Actually, when I look at the targets on the whole, there are actually less targets identified when you do when you measure the co-cultures. From my thought process, it's probably due to interferences. And yes, there is a loss of information from the shared peptides. I have tried trying to add them back in, it just becomes difficult because you really can't tell what was the parent protein of those shared peptides. So it actually really shrinks the fold changes in some of the math that I've done. So and
Yeah.
in other papers, they've also with in particular co-culturing with these two species, yes, with high similarity is very, very difficult on the analysis side because yes, there is the chance that it is more difficult to then parse out what to do with the shared peptides.
Okay. Maybe I just have one real quick question. Do you have any hypotheses about why in mouse versus human you see kind of the different expression of the UPR kind of components?
Yeah, we thought a lot about basically, I mean, it's kind of interesting because the proteostasis network has been shown in other model systems to decline with age. It's kind of interesting here because is it that the glia are somehow compensating for the neurons that's why we see that kind of increase in some of these proteostasis factors. But as far as the neurons, it kind of seems as though something maybe with age, this is one of the things that we're testing and trying to get at in some challenging follow-up experiments, where basically is there something where even when you are trying to activate the UPR that something's going on as far as like on the more of the genomic side where we can't actually these transcription factors can't access the DNA or something like that. kind of remains to be, or just is there just a general decline in proteostasis with age? So those are kind of my thoughts on that. Not sure if that answers your
Yeah.
question, but
Oh,
a
cool.
couple things that I've been thinking about.
Yeah. All right. Well, great. Thank
Thank you.
you,
Thank
Leah,
you.
for
Sorry.
really nice
Thank you
talk.
so much. Thanks.
And we'll just kind of keep moving right along. And so our next speaker is Corey Matsumoto, and he's from UIC. I'm assuming that's University of Illinois, Chicago, or is that a different UIC?
Yeah, that's the UIC.
All right, great. And so I will just turn it over to Corey
Or it's the, yeah, University of Illinois is at Chicago.
at
Yeah.
Chicago. Yeah.
Okay. So can you see this screen fine?
Yes.
Looks great.
Okay. So my name is Corey Matsumoto from the Gao Lab at UIC. And so I'll be presenting the automated containerless sample processing system for single cell multiomics. So to introduce single cell analyses, I want to introduce the importance. So one of the main reasons we do this is to identify cellular heterogeneity. That cells we might think are the same, but when we take a closer look at the analytes, we can actually differentiate them. In cancer biology, for example, this can be important when you're trying to identify any rare cell types as well as any rare cancer subtypes. So now one of the limitations to single cell analyses is that there's not a lot of sample to a single cell. It's been reported that a single HeLa cell only has about 300 picograms of protein, 15 picograms of DNA, and 30 picograms of RNA. So not a lot of sample to start out with. And this proposes an even more of a challenge when you consider that you can lose a majority of the sample to container binding. So now to further emphasize the issue of container binding, I'd like to show this paper in our lab by Dr. Ziwei Zhang and Dr. Gao, where they tested the amount of BSA protein binding to various tubes, as you can see on the x-axis, using various amounts, 1 microgram as well as 100 nanograms. What we could see from these results is that as that we go to lower amounts, in particular 100 nanograms, we see a significant increase in the amount of loss. And you can think of a single cell having 300 picograms, this loss is going to be even more significant. But not only the loss do we have to consider, we also have to consider that depending on what container we use can result in different amount of protein loss. And so these are things we have to take into consideration when we're doing single cell analyses. So now different single cell techniques utilize different methods to reduce this amount of sample loss, such as SOPMS, which utilizes a surfactant, or nanopots, which utilizes very low volumes as well as specialized containers. However, pretty much any current single cell technique utilizes some sort of interface that you can lose your sample. So now acoustic levitation proposes to be a solution by processing everything mid-air and without a container, just using sound pressure. However, many different acoustic levitation platforms aren't that stable when it comes to levitating aqueous liquid droplets, such as this paper featured in Nature, where they were only able to levitate a droplet for about 7 to 15 minutes. And so we really need to develop a more stable acoustic levitation platform. And so that introduces the LevCell, a fully automated containerless sample processing system, which utilizes acoustic levitation and is developed in the Gao lab. So this is the main body of the LevCell. It's comprised of 3 main components. First, we have our transducer base in blue. Now, transducers are ultrasonic speakers, which help produce the sound pressure necessary to levitate our droplets. And what you could see from this illustration is that it's in a bowl-like array. So what this array does is it allows the stabilization of the droplet from multiple different angles, as well as it's more resistant to changes in temperature as well as humidity, which is important when you're trying to replicate the experimental settings needed to perform your experiments. We also have a camera for visualization, as well as a temperature-controlled humidifier to maintain a stable droplet size as well as reaction temperature. Now, more importantly, this system is a smart system, meaning that the video of these levitated droplets is fed through this camera to an SoC, which is in our case is a Raspberry Pi, which processes this video and identifies these droplets. And based off of the droplet identifications, it can autonomously determine and change the different settings, being the heat, the humidity, as well as the voltage. And we'll see how that comes into play in the later slides. And so essentially, with the system, we have stable droplet levitation. So much so that we can do a cell viability assessment of K562, where we incubated cells in a levitated droplet for up to 6 hours. In particular, for our experiment, we found that at 6 hours, it was about 96% viable. And so an experiment we are currently doing is we're trying to incubate the K562 for in excess of 24 hours to see if we can potentially culture cells in a levitated droplet. And that's because with our stable system, we're able to achieve reaction times of over 24 hours. So now let's get into the main components of the left cell. We have the main body, the automated arm, as well as the single cell isolation system. So let's focus on the first two. And to do that, here is a video with the main body to the left and the automated arm to the right. And so if we take a closer look at the main body, on the top, we have our SoC, which is our Raspberry Pi, which processes all the settings and controls it autonomously. We have the transducer base in blue, the camera in red, and the temperature-controlled humidifier to the left. Now, if we take a look at the automated arm, we have 4 different arms for sample addition, cell addition, or sample aspiration, followed by 4 different syringe pumps in the back. So now let's take a look at what it looks like from the eyes of the camera. So here we have the automated arm adding 5 droplets in parallel. So these can be 5 individual cells. And so utilizing computer vision, this is what it looks like from the eyes of the left cell. You can see that we're able to easily identify the different contours of these droplets. And so from these contours, it's able to draw these different boundaries. And from these boundaries, we get 2 types of important information. One is the volume to the right, as well as the shape and size, which is the width to height ratio, being one, as you can see to the left. Now, the volume is important because if this droplet is becoming too large, the left cell can recognize this and autonomously reduce the output of the humidifier. If it's too small, the left cell will autonomously increase the output of the humidifier just to maintain that stable droplet size. Now, the width to height ratio is important because this relates to voltage. And that's because if you have too high of a voltage, the shape will compress until you get this droplet bursting. Too low voltage, the shape will become more spherical and you get your droplet loss again. And so this represents what's happening during your incubation steps. If you had an 8-hour trypsin digestion, for example, throughout that 8 hours, the left cell will continuously monitor and identify these levitated droplets and then diagnose and automatically adjust the different variables, being the voltage and the humidity, as well as constantly monitoring the temperature. As you can see in this case, it's held constant at a temperature of 30 degrees Celsius. So after our incubation steps, we'll get addition of reagents, and this is actually contactless addition. So a reagent droplet forms, which only touches that sample droplet. The capillary never touches. So this is to prevent cross-contamination as well as any loss to that capillary tip. And so we do this by identifying and mapping out the precise coordinates of that capillary tip as well as the levitated droplet utilizing computer vision, as you can see to the right. And so after an incubation step, we'll get sample aspiration to the LCMS for analysis. You can also get an additional reagent addition because we have that extra arm, but this just shows the sample aspiration. So now to further implement automation as well as improve our throughput, we also custom built a single cell isolation system. So this is the basic schematic of how it works. And in our system, there are 2 different scenarios. 1, we have multiple or no cells, which we don't want to isolate. And 2, we have a single cell, which we do want to isolate. And so how it works is we load a cell solution to a custom built syringe pump, which flows these cells through a capillary, which is visible by a microscope camera, which is also connected to the same Raspberry Pi as the love cell. So utilizing computer vision, it's able to interpret how many cells there are. In this case, there are multiple cells. And so there will be a downstream 6-port valve, which will direct this to waste. Now let's take a look at the single cell scenario. And so the Lev cell will be able to identify autonomously that there is indeed a single cell in this capillary at a set time. So it'll autonomously switch this 6-port valve in line with this automated arm, followed by another switching back step, so that a syringe pump filled with water or buffer can continue to flow the cell through this microscope viewing area to the automated arm and eventually to your levitated droplet. And so let's take a look at what it looks like through the eyes of the microscope camera. So here we have the top capillary leading to the 6-port valve, and here we have cells actually traveling through the capillary. And so this is what it looks like from the eyes of the Raspberry Pi. Essentially, you can see we can easily identify the boundaries of the different cells. And from these boundaries, it's able to interpret if it's no cells, multiple cells, single cells, or even debris. As you can see, this is likely debris because it's too small to be a cell. And so let's say that there is a single cell moving through this capillary at a set time. The Lev cell will autonomously take an image, and from this image, you get two types of information, one being the shape of the cell as well as the size. As you can see, this cell in particular is about 14.4 microns in diameter. Now, this can be important when you're trying to relate the physical properties to downstream analyses. For example, one cell, which is a lot bigger, might have a lot more protein compared to a cell that is smaller. And so it'll automatically store this data. And so to ensure that the cell is moving through the 6-port valve through the bottom capillary to the automated arm, there will also be output video recorded, where in this case, indeed, we see that there is only one cell moving toward the automated arm and eventually to your levitated droplet. And so by implementation of all these three different processes, our single cell isolation system, our automated arm, and our main body, we're able to achieve fully automated multi-omic single cell processing. That's assessing RNA, proteins, metabolites, and lipids from the same single cell. And so this is a basic schematic of how it works. So out of these analytes, we first need to extract RNA, and that's because RNAs are prone to degradation through RNases, as well as it's the peptides, metabolites, and lipids that can all be assessed together through LCMS compared to the RNA. And so here are results from real samples that are processed in the Lev cell. Just for full transparency, this transcriptomics data is from 100 cells, and that's because we're currently still working with different single cells RNA sequencing kits to get down to that level of sensitivity. But to extract the RNA, we're currently exploring two different avenues. First, you can see here on this slide is a magnetic bead RNA extraction. And so we add magnetic beads, which will bind to RNA. And after an incubation period, we add a magnetized steel rod, which is connected to the automated arm. And we touch the tip of this droplet, which will organize or isolate the magnetic beads to the tip, which we can then extract for downstream analysis. However, we are also experimenting with just taking a 20% aliquot from this droplet, leaving the remaining 80 for proteomics, metabolomics, and lipidomics analyses. So other than the transcriptomics data that you see here, it's important to note that the proteomics, metabolomics, and lipidomics data are from real single cells processed in the Lev cell. And so what we did is we compared 50 single cells processed in the Lev cell versus 50 single cells processed in the tube. If we take a look at the proteomics data in terms of protein and peptide intensities as well as identifications, the Lev cell process samples outperformed the tube process samples. What this means is that we're assessing more analytes, resulting in greater intensity and more identifications because we're not losing anything to tube binding. Now, we also saw from the same samples that we got this data, by optimizing the LCMS, we can actually identify additional metabolites and lipids. And so the metabolites shown here are the different amino acids. However, we also identified various other metabolites. And so how we verified these amino acids were based off the precursor ion mass, as well as correlation between the order of the experimental retention times with the reported tension coefficients from another paper. In addition, we also identified various lipids. This is just one out of many, which we identified, LPE18. How we verified them are based off, again, of precursor ion mass, as well as we looked at the MS2 fragmentation data to see if there was any signature ions which were generated, just to further ensure that indeed we are identifying this lipid. Now, it's not shown here, but it's important to note that the lipids and metabolites identified in the Lev cell process samples had significantly greater intensities compared to those in the tube. And that again is because we don't have a surface to lose our sample. So to summarize, the LevCell is the first containerless cell processing system. The LevCell implements autonomous functionality to ensure droplet stability. That just goes back to the implementation of computer vision, allowing us to levitate a droplet for upwards of 24 hours. Compared to tube samples, the LevCell cell shows improvements in peptide, protein, lipid, and metabolite abundances, and we're still working on the transcriptomics data to get that down to a single cell level. And finally, through implementation of automation and the ability to process multiple samples at once, we can achieve that necessary throughput for single cell studies. This goes back to how for each Lev cell, you can process 5, which was shown to up to 7 droplets. Not only that, but these systems are very cost efficient and compact. So you can have multiple these different systems within the same lab to achieve a higher throughput. With that, I would like to thank the many members of the GAL Lab as well as our collaborators. I'd like to thank you for listening and take any questions.
Great. Nice talk, Corey. Do we have any questions? Please use the chats or the question and answer for those of you online. I could maybe start off while people might be typing.
Yeah.
And so I'm just kind of curious, I don't know much about, the physical process of levitating the cells. And have you or others looked into what kind of stress that might induce into the cell? Like, would you expect to see a little bit of cellular changes because of the pressure that you're putting on the cells? Is that something that you plan to do in the future once this is fully established, to really kind of compare this to like other, potential single cell approaches and see if there might be something there with regards to like alterations of proteomes or metabolomes?
Yeah, so that study currently hasn't been done. And essentially, when you take a look, or when you consider the acoustic pressure that is being created to levitate your droplets, essentially you have a very high pressure acoustic pressure under the droplet. And at your actual droplet, it's very low acoustic pressure. So they don't experience much acoustic forces. There is some other forces you have to consider, such as there's spinning internal forces inside the droplet.
Okay.
But I think that's something we can definitely do with our system, especially at a single cell level to measure if there's any perturbations or things like that.
Yeah. Looks like there's one question on the Q&A. I'll go ahead and read it off. So, what kind of variations in proteomes, transcriptomes do you get between cells of the same treatment? Also, how many cells can you process per day?
Oh, that's a good question. So, in terms of we don't, or currently I didn't look at any differences or in-depth differences between the cells processed in the tube versus the lab cell. All we did in this analysis was just really compare the identifications. But I think that goes back to the previous question where there can be differences in acoustically levitated cell. And so that's something we might need to consider looking at in the future. In terms of the throughput, it really depends on your mass spec at that point because we're able to fully saturate a mass spec one, whether it's it could be 5 minutes. And so what we think is that with our throughputs, we can get more than 100 cells per day, but it really depends on the output of your mass spec because we're able to saturate the machine.
And maybe one more quick question. This is really interesting technology. Could you culture enough material to measure growth of 3D culture systems under distinct treatment?
So I'm not very familiar with
The 3D culture.
the 3D culturing.
Yeah, I guess they're maybe asking kind of the opposite of doing single cell, but could you get enough cells that it would turn into a kind of a 3D culture system
Yeah,
with your...
yeah. Well, I'm not sure exactly like the turnover time to form those 3D culture systems, but we can levitate cells for longer than 24 hours, meaning that we can have at least one turnover. Uh, but again, we haven't really gone through that initial tests where we're actually seeing if they actually grow in the levitated droplet. That's what we're currently doing.
Yeah. I think some of the, yeah, I mean, for some of the 3D culture systems, they might need more than 24 hours, but
Yeah.
you know,
I mean, with our system, I think we could go well beyond 24 hours.
okay.
So, yeah.
All right, cool. Well, thank you, Corey. Very interesting talk. Cool
Thank you.
technology that's coming up. Our last speaker for this webinar is Isaac Plutzer, who's from the Washington University School of Medicine in St. Louis. And so, Isaac, turn it over to you.
Hi, thank you for the introduction. So yeah, I'm Isaac Plutzer. I'm a grad student in Ben Major's lab at Washington University in St. Louis, and I'm a fifth year in the computational and systems biology program here. Today I'll talk to you about how I'm using proximity labeling and phosphoproteomics to illuminate the dark kinome. So I study protein phosphorylation as a mechanism of cell signal propagation. About 3/4 of the human proteome is phosphorylated, and this post-translational modification affects the stability, activity, localization, and physical interactions of substrate proteins to regulate their function. Phosphorylation is carried out by a family of proteins known as the kinases, which we say are highly druggable because they require binding of ATP in order to function, and that can be very effectively inhibited by blocking the ATP binding site. So in this kinome tree on the right, I'm showing in red kinases that have FDA-approved selective small molecule inhibitors that are being used to treat a variety of diseases. However, a larger subset of the kinases that I'm now showing in blue are what we call the dark kinases, meaning that little is known about their function and we don't really know what their substrates are. And so there's a considerable push in the field right now to identify the functions of these dark kinases and pick out those that have therapeutic potential. And so the way that my lab is doing this is to use proximity labeling, which allows us to define the cellular neighborhood of each of these kinases and identify what cell structures or protein complexes a kinase is part of. The way we do this is we take a kinase of interest that we also call the bait protein and fuse it to a promiscuous biotin ligase here called mini-turbo and stably express this in a model cell line. When we add exogenous biotin into these cells, the proximity labeling enzyme creates a zone of highly reactive biotin AMP molecules that irreversibly and covalently modify surface-exposed lysine residues on nearby proteins. We can then purify these proteins with streptavidin and analyze and quantify them by mass spectrometry. So the data collection for this in our lab has largely been done by a research instructor, Dhaval Bhatt, who's generated hundreds of these cell lines, done hundreds of these pull downs and hundreds of mass spec runs. And so I've joined this project as kind of a data analyst to make sense of what this data means in picking out functions for these kinases. One main challenge is that these streptavidin purifications capture a lot of non-specific protein interactions. So many proteins will bind the streptavidin, as well as kind of non-interacting proteins that by random chance happen to float through this labeling zone. And so if you look at the raw data coming off the mass spec, the proximity network would look something like this. So a large part of my work has been to develop methods to filter this data and reduce the network down to something more biologically meaningful. And so to give a short summary of how I do this, we do these experiments with a variety of controls, including the parental cell line and cells expressing just the biotin ligase on its own, localized to different parts of the cell. And so I can then score each of these interactions using different algorithms. The first that I'll talk about is the SAINT score, which you can think of as a direct comparison to negative controls. And then another score that I've been using is the COMPASS WD score, which is a comparison to the rest of the data and kind of gives a boost to proteins that uniquely interact with one or a handful of kinases. I can evaluate these methods using ROC analysis against databases of known protein-protein interactions like the BioGrid database. And so this ROC curve shows how accurate each of these scores are at predicting known protein-protein interactions and essentially filtering out the true positives from the non-specific interactions that we capture. And so what I'll point out is that each of these scores kind of on their own is kind of modestly predictive of these true protein-protein interactions. But one thing we've seen is that by combining these scores, combining this direct comparison to a negative control, as well as this comparison to the rest of the data, we can achieve much better kind of predictions of these known protein-protein interactions. And so that allows me to take those big jumbled networks that I kind of showed before, like this, and reduce it down to something that is more meaningful, tractable for our analysis. These smaller, more curated networks allow us to kind of annotate each of these interacting proteins and begin to understand the function of this kinase. So for this kinase BRISC2, I can highlight the localizations of each of these interacting proteins and begin to piece out where this kinase is in the cell. One thing that this data doesn't provide us with, however, are an identification of what the substrates are. So we get these big networks of, lots of interacting proteins within the cell. But the next question is, which of these are the substrates? And so this is also a kind of major problem in the field of phosphoproteomics, in that most phosphorylation sites that we've seen by mass spec don't have a known kinase that's responsible for that phosphorylation event. This review kind of highlights it pretty well, where they show a kinome tree and the size of the circles are representing the number of known substrates for each kinase. And what I think you can appreciate is that there's substantial bias in these annotations. The vast majority of phosphosites that have been annotated belong to a small minority of well-studied kinases. And so kind of second branch of my thesis work is to develop methods that can identify substrates in an unbiased way. And so the way that I've worked on doing this is to use transient overexpression, gain of function type experiments, followed by phosphoproteomic profiling. So with this workflow, I take a kinase of interest. Here I'm showing BRISC2, and I transiently express it in a model cell line along with controls, either a transfection control or a kinase dead mutant. Following overexpression of the kinase, this leads to a rapid increase in phosphorylation of the substrates. And then I'll do a phosphoenrichment and deep profiling of the phosphoproteome using TMT labeling and offline fractionation to identify changes in the phosphoproteome that occur following overexpression of the kinase. And so the data for this experiment will look something like this. You can see that following overexpression of the kinase, we get a substantial induction or upregulation of phosphorylation sites. But the problem is that a lot of these sites may be downstream substrates, not necessarily directly phosphorylated by BRISC2, but further down in the pathway. But what I can do is I can map each of these sites back to our proximity data and identify sites that are both upregulated following overexpression of the kinase and proximal to the kinase in the same part of the cell at the same time. And this allows me to filter down this list of changing sites to a smaller, more curated list of likely substrates for the kinase. So again, showing this network, this allows me then to annotate within this network which are the likely substrates of the kinase. The other thing that I can do from this phosphoproteomics data is begin to analyze cell pathways that are regulated by the kinase. So here I'm showing PTM enrichment analysis, which is very similar to a gene set enrichment analysis, where I'm looking at known lists of phosphorylation sites for different cell perturbations and comparing it against the data that I saw. And this analysis in particular picks out different signaling pathways that appear to be upregulated or downregulated. And what I can do is I can go into lab and test this. So from this analysis, it predicts upregulation of AMPK. And so I go into lab and I can do a Western blot for AMPK substrates. Here I'm comparing the wild type kinase to transfection with the kinase dead control. And what you can see is that there's an increase in signal from AMPK consensus sites, phosphorylated AMPK consensus sites with the wild type kinase when compared to the negative control. This allows me to kind of begin placing this kinase within known signaling pathways. And then what I can do is I can kind of follow these signaling pathways down. So I can go down the list and now look at mTOR signaling. In our PTM enrichment analysis, we see a decrease, downregulation of mTOR signaling. And so here I'm plotting for serine 2448 on mTOR, and it doesn't look quite as good, but we can see a slight kind of reduction in the phosphorylated active form of mTOR. And then I can go a step further and look at the substrates of mTOR, showing a decrease in the phosphorylation of these substrates. Here showing S6K, and then again for 4-EBP1. So this allows us to kind of take an unbiased, holistic look at different kinases and begin to place them within cell signaling pathways. So to summarize here, if I can change my slide is stuck. There we go. So to summarize, we're using two different techniques, proximity labeling and phosphoproteomics. The proximity labeling allows me to identify physically where in the cell a kinase is localized to. And then with the phosphoproteomics, I can place that kinase within known signaling pathways and identify kind of landmark substrates for the kinase. In my ongoing work, I'm using our proximity data to identify kinases that are most likely relevant to cancer signaling pathways and prioritizing phospho-analysis of those kinases. And so with that, I'll wrap up this talk. Highlight people in my lab that have done a lot of this work. Dhaval, I mentioned before, is a staff scientist who's done a lot of the proximity labeling experiments. And Emily is another senior research scientist in our lab that has helped me with kind of everything mass spec. So with that, I'll take questions. And thank you for the time. Thank you.
All right, thanks, Isaac. Again, if anybody has any questions out there, please kind of type them in the question and answer chat. I'll just go ahead and kind of start. And so you find a lot of proteins that are not phosphorylated, but are being pulled down by your proximity approaches. Is there anything in the data set that you can look at to give you an idea whether they're still direct interactors of your kinase, but maybe not necessarily substrates, but maybe they're like scaffolding proteins that, you know, complex with another protein that gets phosphorylated? Is there anything that you could possibly kind of tease out to continue to determine whether a protein is still is a direct interactor versus some kind of secondary protein?
Yeah, I mean, from our data, the data detects proximity. So it's hard to parse out whether that is a direct physical interaction or a more transient interaction, or like you mentioned, an interaction that's mediated by another protein. In our kind of constructs we make, we have a V5 tag, so we can validate some of these things by IP mass spec.
Yeah, that's what I was thinking too.
More of a direct physical interaction. But I think one kind of interesting direction that we're kind of going in is to look at kind of known protein binding domains on some of these scaffolding proteins. So from this proximity data, we can test for enrichment of particular protein domains. And one thing that we get a lot are, again, these scaffolding proteins, protein binding domains. And so one thing that I think we can gain from this data is kind of understanding which binding domains on scaffolding proteins can fit the different kinases that we're interested in.
Okay, cool. Yeah, several questions popped up in the chat. I'll just read them off. Do you plan to use mass spec to measure differential abundance of phosphorylated substrates? The Western blot quantification seems difficult.
Yeah, that's a great question. Yeah, so with the mass spec, even with the TMT labeling, deep fractionation, we don't see some important substrates. I think the ones that are often interesting are very low abundance proteins, and that's why I'm using Western blot to see some of these mTOR substrates. I think, I mean, I haven't really seen many kind of phosphoanalyses that can see that deep, but I think maybe there's other ways we can do that. Our lab has been using SureQuant for some of these low abundance substrates. That's a possibility. I've seen labs kind of doing hybrid DIA phosphoproteomics methods. So those are things
like a PRM type of method for specific. Yeah. Another question in the chat. I know some kinases change their subcellular localization based on treatment signaling. How would your approach address this? And then also, do you think you could determine temporal regulation of localization, or is the timing not able to be that precise?
That's a good question. I've been wanting to kind of do kind of time course experiments with both these proximity labeling constructs and the phosphoproteomics, but it's not something we've really tried yet. Yeah, I mean, it is something we could do. I've been wanting to, for example, you could overexpress a kinase and then do, proximity labeling for a downstream kinase and see if it changes localization, or treat with a drug for known signaling pathways. It's just not something we've quite tried yet, but I think it's a great idea, a good potential future direction for this research.
Yeah. And maybe one more question. Lovely talk. Are you using automated bead-based phospho enrichment? What's the minimum number of baits you would need for a specific compartment in order to be statistically significant? And what if you're looking at a condition or stress where there is no stress-specific database of known interactions? Good questions.
So yeah, thanks for all those questions. To kind of go through them in order, we recently got a Kingfisher machine that now lets us do the automated magnetic bead-based enrichment, and so I'm now using that. Some of the BRIS2 data I showed today, that I did by hand, but the bead-based enrichment has been working a lot better. It gives better data and is just kind of faster overall. For number 2, minimum number of baits for a specific compartment to be statistically significant. Yeah, this is not something that we've kind of fully fleshed out yet. So we have control baits that localize. We have NLS, NES, so cytoplasm and nucleus. We have an ER control bait, a plasma membrane control bait, and a mitochondrial bait. And so the difficulty has been that we have kind of mixed results. Some of them work well, some of them don't work as well. And I think it kind of depends on the bait protein and the compartment it's in. For Saint analysis, you really just need one good control to compare with your experimental bait. And so for that, everything works well, kind of except the mitochondria. The mitochondria just seems like such a small compartment that everything gets labeled. And so it's hard to do the statistics for baits that localize to that compartment. For the other score, the WD score, where you're comparing across the full data set, I'd say it helps to have at least 5 to 10 different baits to really get the most out of that score. But that again will depend on how different the baits are, whether they're in different parts of the cell or the same part of the cell. So I'm not sure I have a great answer for that for like within the same compartment. And then then the third question is a really good point. When we do the PTM enrichment analysis, we're relying on kind of previously published data on certain cell perturbations in order to make these assertions. And so if we don't have data for one of those kind of signaling pathways, it won't come up in our screen. And so that's definitely a limitation of that analysis. There's just not quite as much data, phosphoproteomics data out there to do really deep analysis on some of the less well-studied signaling pathways.
Okay, great. Well, just want to thank Isaac one more time, and also all the other speakers as well. It's just really great to hear your research and what you guys are up to. Thanks for sharing your highlights with us. And thanks to everyone out there for tuning in. And I guess we'll see you at the next US HUPO webinar. Thank you. I think we'll get going. So